Hi, I'm Steve Mayfield. I am professor of biology here at UC San Diego, and I'm also the director of the California Center for Algae Biotechnology. And today I'm going to tell you a little bit about why we think about using green algae for the production of biofuels or some other high-value bioproducts. So with algae, we have a couple of different choices, and you've already heard, I think, or you will hear in other parts of this MOOC, about cyanobacteria and also about diatoms, and today I'm going to tell you about green algae. So algae is a very diverse set of organisms that span very far on the phylogenetic tree. And although phylogenetic trees are a little hard to understand, let me just show you these two right here. These two lines right here are human beings and worms. And the distance they are apart tells you how far they have, how many years they've been separated in evolution. So worms and humans pretty far. Here's red algae, here's green algae, here's cyanobacteria. So these span billions of years in evolution. And what does that mean when we say billions of years in evolution? That means that the genetic diversity inside those organisms is really great. They are really different from one another. But they all do one thing in common, and that's photosynthesis. Okay? So when we talk about what algae we might use, we can think about cyanobacteria, we can think about brown algae, which are diatoms, or we can think about green algae. But any one of those has to satisfy these four criteria. So first of all, they have to be scalable. And what does that mean? We've talked about this many times, but in fuel, 1.2 trillion gallons per year of fuel are consumed on this planet. In the United States, 300 billion. Obviously, they have to be productive. It doesn't do us any good if we're going to turn all of our available land, now that we maybe grow crops or that we have forests or that we have something else into, it doesn't do us any good if we're going to turn all that into making biofuel. So the higher the productivity, the smaller the footprint. Sustainability, that means that we can't think about using up all of our water or all of our nitrogen and phosphate. We need those for food production. So sustainability in terms of biofuel production in algae means we have to use non-arable land, non-potable water. And then fungible fuels. What does fungible fuel mean? That means it's a drop-in fuel. There's $13 trillion of existing infrastructure in the United States for fossil fuel, the refineries, the cars, the gas stations, you name it. So it really helps a lot if we can make a fuel that drops right into those so we don't have to change any of that stuff. And as I'll show you, algae biofuels fit that criteria. So first of all, productivity. There's several ways to measure productivity. I think the easiest way to think about this is in terms of tons per acre per year or kilograms per hectare per year. Algae right now today, we have algae that will hit 50 tons per acre per year. The most successful terrestrial crop, so the best higher plant is sugarcane. It gets about half that. It gets about 25 tons per acre per year. You can read that sugarcane can have much higher yields than that, but that's generally because they report that with water in the sugarcane, because that's the way it's sold. So when you convert that to dry tons per acre per year, algae is the best. Another way you can look at this is in terms of oil content. Here's several different crops that we use today, either as food oils or we turn them into biodiesel, things like canola and safflower and soy, and I think you'll hear later about jatropha. So all of these have a pretty good oil content, and algae's right up there with the best of them, anywhere from 20 to 60%. But because its biomass yield is so high and it has such high oil content, we can actually hit numbers of something like 5,000 gallons per acre per year. So what does that mean? Well, here's one quick math on that. 5,000 gallons per acre per year times 60 million acres would give us 300 billion gallons. So that would be 100% of the petroleum we use in the United States. So 60 million acres seems like a big number. In the United States, we have about 100 million acres of corn. We have about 56 million acres of soybean. We have about 50 million acres of wheat. So these are numbers that we already have reached in agricultural production. In fact, in the United States, we take 40% of our corn crop, so that's about 40 million acres of that, and we convert that 40 million acres into only about 13 billion gallons of fuel. So if we took that same equivalent land that we're using for corn right now and put that into algae, we'd already get an enormous improvement. This is just some of the oil content of some different algae, and some of them can be very high. You have things like Botryococcus here, which can hit 75% of its dry weight as oil. But all of these are very good. All of these things are in the range of 20 to 50%. Certainly high enough range that they could be economically viable. So what does the process look like? These are actually pictures from our greenhouse here in San Diego. At commercial scale, it'll look a little different, and we'll show you that in another part of the MOOC. But here's algae growing in hanging bags in one of our greenhouses. You concentrate the algae down, so you simply spin the algae down and concentrate it. You then separate it into the oil component and into the carbohydrate and sugar component, and then the oil component gets concentrated, and that can go directly into traditional oil refineries. And of course, the reason it can do that is because the petroleum we pull out of the ground, in fact, is fossil ancient algae. So this is the way the process looks, right? All aspects of this have been worked out, and in fact, fungible fuels have already been made from algae. Sapphire Energy, a company that I'm a founder of, and I think you heard Rob McBride talk here, they have a little car called the Algaeus, which they drive around on fuel made 100% from algae. They also blended some algae fuel into a jet aircraft and flew it. And then another algae company called Solazyme, and they produce their fuel in a little bit different way. They do it by heterotrophic growth, so they actually feed sugar to the algae and have it converted to the oil. They produced biodiesel, and that deal was used to drive this amphibious vehicle. And then also in the summer of 2012, some of that fuel was put into U.S. naval boats and driven up and down the coast here. Okay? So if it all works and the process works, why are we not able to buy this in the stores right now? It's because of the cost. So it is still too expensive for the fuel we produce in algae now to be competitive with fossil fuel. This is an analysis we did several years ago in 2009. At that time, we thought the cost might be around $21 a gallon. And we said just by bioprospecting, so just by going out and getting strains that grow to higher biomass per acre per year and have higher oil content, we should be able to get that cost down to about $8 a gallon by 2012. And sure enough, we accomplished that. The era we're in right now is in here where we're looking at engineering. So how do we get the ponds cheaper? How do we get harvesting cheaper? How do we get CO2 delivery cheaper? This is all going on right now, and this will drive the price of that down, we think, over the next 4 or 5 years to be something around $4 or $5 a barrel, $4 or $5 a gallon, sorry. And then the final thing that we have to do is we have to get co-products. So we have to do something with the non-fuel component of algae. In agriculture, there's an expression in pig farming, you have to sell everything but the oink in order to be profitable, and we have to do the same thing in algae. So I want to talk just a little bit now about some of the co-products that we might make and how we might make those co-products. So one of the ways we think about that, and specifically in my lab, is we think about doing transformation, so introducing a foreign gene into the algae that will produce a very high-value protein. We can then extract that high-value protein, sell it, and then the rest of the algae can be used to make oil or make other components that we want of it. So when we think about putting proteins into this, we have 2 places we can put them. We can put them into the chloroplast genome, or we can put them into the nuclear genome. Both of these genomes contain genes. If you put a gene in there, the protein that's made from it is made in, obviously if it's in the chloroplast genome, the protein is made here. If it's made in the nuclear genome, the protein is translated in the cytoplasm, and then that protein can be either secreted out of the cell or targeted into the chloroplast. The chloroplast has a relatively simple genome. It's a little circular genome. It's about 200 kilobases inside of Chlamydomonas in the green algae we work with. Only encodes about 40 different proteins, pretty simple promoters. Transformation goes by homologous recombination. So this is a pretty simple system. And in this system, we've already got very high expression of lots of different recombinant proteins. So this is simply a protein stain gel of wild-type cells. Here are 2 transgenic lines, and these happen to be expressing a recombinant protein from bovine, from cows, called serum amyloid A3 or mammary-associated amyloid. And what this protein does is when you eat it, any mammal, when they eat this, the protein passes through your stomach, gets into your gut, and stimulates mucus production in your gut, and that gives you a physical barrier that protects you from bacterial and viral infections. So very valuable protein, and one we can certainly think about making, and there are many, many others that we can make. For nuclear transformation, it's a little more complex, random integration. We have much more complex promoters, but we can target proteins to different parts of the cell, and so it's much more powerful. But a big problem in all algae has been that nuclear gene expressions are often poorly expressed or silenced. And so we've spent several years working on that, and recently we've been successful in getting high levels of expression. And here's just some that I show you right here. So we picked 6 different fluorescent proteins. So here's one called blue fluorescent proteins, cerulean, green, Venus, tomato, and cherry. And these are algae that are expressing that recombinant protein. And the way this recombinant protein works is when you shine light on it, it absorbs that light and then fluoresces the light back out at a different color. And as you can see, we can make these beautiful fluorescent algae. Now, those are not a valuable co-product in themselves, but what they show you, I think, that if you can make rainbow-colored algae, you can make just about anything, right? More important than that, we can target these proteins to different parts of the cell. So in this case, we targeted the M-cherry protein to the endoplasmic reticulum, so that's the part of the cell that secretes proteins out. Here we put a blue protein into the nucleus. Then we cross these 2 strains together, so we have red endoplasmic reticulum and a blue nucleus. And then we cross that cell with another cell line that's expressing green fluorescent protein in the mitochondria, and that gives us this really beautiful thing, right? So blue nucleus, red ER, green mitochondria, and then finally we cross that with another strain that we had put a blue fluorescent protein onto the flagella, and we end up with this really beautiful rainbow-colored algae. Now, this doesn't have any particular value on itself, but I think what this really nicely demonstrates that all of the genetic tools that have traditionally been used in higher plants, so that's transformation, that's breeding, that's selection, that's all of those things, we can now use these to impact algae, to really drive the products that we want to make, but at the same time we can also drive higher fuel, higher lipid accumulation, higher fuel accumulation. We can get more productive, we can put crop protection. I believe the era now of sort of synthetic biology has really arrived. So what does that mean? What that really means is that we are now poised to do the domestication of algae. And this is just an example that I always like to show the students. So this is domestication of corn. So 7,000 years ago, corn looked like this little scrawny guy called teosinte. And by breeding and selection, over 7,000 years, we turned this little guy into this big beauty right here. This big beauty keeps 7 billion people on the planet fed. I mean, we did the same thing with wheat, we did the same thing with rice, all of them. We had this domestication which greatly enhanced the ability of these organisms to be productive. And we need to do the same thing in algae, and we are poised to do it now. So I think what you'll see over the next couple of years is improvement in the genetics, improvement in our selection, improvement in our breeding. We're already at a basal level that's pretty good, so we can produce about 30 tons per acre per year of algae, and that can be about 30% lipid. I think over the next 3 or 4 years, we'll get that number up to 50 tons per acre and 50% lipid. During that same time, we're going to make improvements in the process, right? Meaning in the engineering systems, in the ponds, in the harvesting, in the extraction, and the conversion of all those. And when the 2 of those things come together, which I think will be in the next 3 to 4 years, then I think we'll have the opportunity to have algae biofuels be competitive with fossil fuels. And of course, as many of you will know, because the fuel is made by photosynthesis, we're capturing CO2 out of the air, so we are recycling CO2. So the net greenhouse gas emissions, much, much lower. So thank you all very much for your attention.